Okay, so our last section is talking about the degradation of amino acids. So we talked a lot about sugars, one section on fatty acids. Now let's talk about degradation of amino acids. So proteins provide energy. If you have no carbohydrates and no lipids available in your body, then we have proteins can be transferred into energy as well. Not as efficiently, but it can be done. So those carbon skeletons of amino acids are obtained from transamination reactions. So we'll talk about that in the next slide or two, and can be used as intermediates in the citric acid cycle to produce reduced coenzymes for energy transport. So we produce ATP through that. So overall, the amino groups on the bottom here, you'll see the amino groups are converted to urea, and that is a process we'll talk about in another video. But overall, the different amino acids kind of have different fates or how they get to the fuel. But these amino acids will all turn into pyruvate eventually, that then can turn into acetyl-CoA. Things like isoleucine, leucine, or tryptophan can be turned straight into acetyl-CoA. And then things like leucine, lysine, and phenylalanine are going to be converted to acetyl-CoA. And those, because we do not have sugars in our system, that triggers ketogenesis. So we're going to make those ketone bodies that can be used as fuel. Okay, so transamination is that process to degrade amino acids and to form those acetyl-CoAs. So in our liver is where our amino acids are degraded. Amino group is transferred to the amino acid in alpha keto acid, so usually alpha ketoglutarate, and it forms a new amino acid and an alpha keto acid. So let's just look at this, for example. So if we take alanine, we have this NH3 here, and something called alpha ketoglutarate, which is formed in our body from glutamate, but we'll see that in a second. So we take alpha ketoglutamate. These two groups are just going to swap places. So that's all that's happening. Okay, so in an alanine, so because we're talking about amino acid alanine, aminotransferase, we're just going to swap those groups. So the alanine forms pyruvate. We've seen this molecule before. Can turn into acetyl-CoA, undergo the citric acid cycle, and then the alpha ketoglutarate forms glutamate. So that is unknown. That's an amino acid we know. So this process happens with a lot of different some of the different simpler amino acids. Okay, then we also have oxidative deamination. So this is just transamination. We're trading where the amine is. Now we're doing oxidative deamination. In oxidative deamination, we remove the ammonium group, so that NH3 plus from glutamate as NH4 plus. So we're going, in our last reaction, we formed glutamate. Now we're going to take that glutamate and transform it into alpha ketoglutarate. So what happens overall? You see this NH3 is oxidized. We change it into an oxygen and then oxidize that up to make that alpha keto. This uses water. That's where the oxygen comes from. Our oxygen there. And then these two hydrogens, one go into forming NADH, so and H plus, which is how we get energy out of this process. So overall, long story short, we're going to use oxidation and transaminations to get to our denitrogenated bodies that then can be broken down even further. So learning check, write the structures for the products of transamination of alpha ketoglutarate by aspartate. Okay, so alpha ketoglutarate is going to trade places with our amine. So this and this are going to trade. That's how we draw these structures. I cheated and went ahead and put the solution down here. So as you can see, again, these two swapped places in order to get our product.